I want you to picture a warehouse for a second.
Okay, a warehouse.
Yeah, and inside this warehouse, there are vats containing roughly 2,500 liters of human urine.
Oh, wow. That is quite an image.
Right, and it gets weirder. This was collected mostly in Japan throughout the 1970s. And that wild juxtaposition, it actually connects one of the most tedious, unglamorous scientific quests in history directly to a medical miracle.
A miracle that, changed hundreds of thousands of lives.
Exactly. And completely by accident, it's the exact same biological mechanism that eventually gave Lance Armstrong the ultimate cheat code to win 7 Tour de France titles.
It really is a stunning through line. I mean, you have this massive, honestly, slightly unsettling collection of waste fluid leading straight to a multi-billion-dollar class of drugs.
Erythropoietin-Stimulating Agents, or ESAs for short.
Right, ESAs. If you've ever heard of epoetin alpha or darbepoetin alpha, these are the medications that totally revolutionized how we treat severe anemia because they essentially command the human body to manufacture red blood cells.
So our mission for today's deep dive isn't just to read off a list of what these drugs do. We are going to explore the actual biological mechanics of how they manipulate your bone marrow.
And we're going to track the really dramatic shift in how doctors actually prescribe them over the years.
Yeah, and uncover a very hard, humbling lesson that modern medicine was forced to learn. We're going to see what happens when the medical community just, assumes that pushing human biology back to a textbook normal is always a good idea.
Because, spoiler alert, it isn't always a good idea. But to really grasp how ESAs became this foundational tool in nephrology and oncology, plus the center of massive international scandals, we do need to go back to that warehouse of urine.
We do. Because finding the natural hormone that these synthetic drugs mimic, that was considered a near impossible task for a long time.
The history here is just incredibly grueling.
It really is. The hero of this origin story is a biochemist at the University of Chicago named Eugene Goldwasser. Beginning in the mid-1950s, he became completely obsessed with purifying this theoretical hormone called erythropoietin.
EPO.
From PO.
Well,
EPO, right. The scientific community knew the body had some kind of chemical messenger controlling red blood cell production, but natural EPO circulates in our blood and urine at just vanishingly low concentrations.
Which totally explains the need for industrial quantities of fluid, right? Goldwasser spent over 2 decades failing to isolate it simply because the body produces such a microscopic amount under normal conditions.
20 years of failure.
Yeah, 20 years. He eventually realized he needed source material from bodies that were in a state of absolute physiological panic.
And that is why much of those 2,500 liters came from patients in Japan suffering from a specific condition called aplastic anemia.
Exactly.
Because in aplastic anemia, the bone marrow is essentially a ghost town, right?
Right. It's failing to produce new blood cells at all.
So the patient's tissues are basically suffocating from a lack of oxygen.
Yeah, and so the kidneys are just screaming for more red blood cells. They're pumping out absolute maximum levels of EPO into the bloodstream, which eventually filters down into the urine.
And even with that highly concentrated source material, after decades of work, Goldwasser's team managed to extract just a few milligrams.
A few milligrams.
Of pure erythropoietin in 1977.
I mean, it was an impossibly small yield for 20 years of work.
Just a few specks in dust, basically.
Yeah, but it provided the genetic blueprint. That tiny purified sample was handed over to a small, newly formed California biotech startup named Amgen.
Amgen.
Yep. And by 1983, a scientist there named Fu Kun Lin managed to sequence the protein and successfully clone the human erythropoietin gene.
And that cloning breakthrough, that changes everything because suddenly you don't need the vats of urine anymore.
Thank goodness.
Amgen figured out how to insert that human gene into the ovary cells of hamsters, of all things.
Right, Chinese hamster ovary cell.
They essentially genetically engineered these mammalian cells to act as microscopic drug factories, just churning out synthetic human EPO.
And that technological leap took erythropoietin from a theoretical, research curiosity to a viable mass-produced drug.
So to understand the impact of that drug, we need to look at the physiology it's hijacking inside you. Because your kidneys are doing a lot more than just filtering waste.
Oh, absolutely. They are incredibly sensitive oxygen meters. They are constantly tasting your blood.
Tasting it.
Well, Yeah, chemically speaking.
Yeah.
Within the renal cortex, there are peritubular cells that monitor oxygen delivery. If oxygen drops, say you've lost blood or you move to a high altitude or your kidneys are physically failing, these cells trigger a genetic pathway called hypoxia-inducible factor.
The HIF pathway.
The HIF pathway,
Yeah.
exactly. It acts like a fire alarm, commanding the kidney to synthesize and release EPO into the bloodstream.
And from the kidney, that EPO travels straight through the circulation and into the bone marrow.
Right, where it seeks out immature red blood cell precursors and binds to specific receptors on their surface.
Okay, so here is where we really need to clear up a massive misconception about what that binding actually achieves. Because when people hear a term like stimulating agent, they naturally picture an accelerator.
Yeah, that's the intuitive jump.
They assume these drugs act like a foreman at a factory, yelling, speed up the assembly line, build these cells faster. But the biology is doing something entirely different.
It is a completely different mechanism, and honestly, understanding it is totally crucial to understanding the drug's dangers later on.
So think of the drug less like a factory accelerator and more like a boss walking into the building and tearing up the pink slips for a bunch of workers who are about to be fired.
That is a great analogy, because inside your bone marrow right now, you have a massive population of early red cell precursors, and their default biological setting is to undergo apoptosis.
Which is programmed cell death.
Right, programmed cell death. Unless they receive a very specific signal to live, they are fated to die within 24 to 48 hours.
Just automatically.
Automatically. But EPO is that survival signal. It is the molecular, When the EPO molecule binds to the receptor on that precursor cell, it completely stops the apoptosis process.
It tells the cell, don't die, the body needs you.
Exactly. That single chemical interaction allows the cell to survive long enough to complete its natural 8 to 10 day maturation process into a fully functioning red blood cell.
And ESAs just exploit this exact pathway.
They do. By injecting a synthetic version of the hormone, we flood those receptors, rescuing millions of precursor cells that the body would have otherwise just allowed to die off.
So when Amgen finally brought Epo-10 alpha to the market in 1989, it was a profound medical breakthrough, particularly for its very first target audience, which was patients with end-stage kidney disease.
Right.
And to appreciate the scale of this victory, we have to look at what dialysis was like before 1989. When your kidneys fail, they stop producing natural EPO entirely.
Which is devastating for blood counts.
Yeah, a healthy person's hemoglobin usually sits between 12 and 16 grams per deciliter.
Right.
But a dialysis patient's hemoglobin would routinely plummet down to 6 or 7.
And just for context, a hemoglobin of 6 means profound, crushing exhaustion. I mean, a patient can barely walk across a room without gasping for breath.
Because there's just no oxygen being delivered.
Exactly. The heart is constantly overworking, beating aggressively just to circulate the tiny amount of oxygen available, which often leads to severe heart failure over time.
And back then, their only recourse was constant chronic blood transfusion.
Which was incredibly dangerous.
Right, especially in the 1980s, right at the height of the AIDS epidemic, before widespread, rigorous blood screening. Getting a transfusion every few weeks carried a terrifying risk of contracting HIV or hepatitis C.
Not to mention, regular transfusions cause severe iron overload, which damages the liver and the heart.
Oh, and they also expose the patient to foreign antibodies, right?
Yeah, which essentially sensitizes their immune system and makes finding a compatible kidney for a future transplant incredibly difficult.
But the arrival of ESAs eliminated that horrific cycle almost overnight. I mean, a simple injection reliably raised a patient's hemoglobin into a safe, manageable range, around 10 to 12 grams per deciliter within just a matter of weeks.
The routine transfusion dependency just completely vanished. It was a flawless example of pharmacological success. You identify a genuine hormone deficiency, and you correct it with a synthetic hormone replacement to eliminate a massive burden of disease.
It was undeniable success. But as we see so often in medicine, it also laid the groundwork for a logical fallacy that eventually killed people.
Yeah, the more is better trap.
Exactly. The medical community looked at the dialysis patients and fell into a trap. If intervening to push a patient's hemoglobin from a dangerous 7 up to a moderate 11 is profoundly beneficial,
then pushing it all the way back to a textbook normal 14 or 15 must be even better.
that was the assumption. And the shifting identity of the drug throughout the 90s and early 2000s is pretty fascinating to trace.
It really is. It quietly morphed from being a targeted hormone replacement for failing kidneys into this broad hemoglobin normalization tool.
Right. Oncologists started using it for chemotherapy-induced anemia. Surgeons prescribed it before elective surgeries.
Doctors began administering it to patients whose kidneys were actually functioning fine simply because they wanted to drive the red blood cell count up to match the numbers of a completely healthy person.
Okay, I have to challenge this logic on behalf of anyone listening right now who might be confused.
Go for it.
If a healthy human body naturally operates perfectly safely at a normal hemoglobin level of 14, why does using a drug to reach that exact same normal state suddenly trigger catastrophic events like heart attacks and strokes? I mean, normal should just be normal, right?
You'd think so, and that paradox baffled the medical community for years. But the answer lies in the physiological difference between endogenous production and artificial stimulation.
Meaning natural versus synthetic.
Right. Achieving a hemoglobin of 14 naturally is a balanced, holistic process. Hitting it artificially requires super physiologic doses of ESAs.
We are talking about massive surges.
Massive synthetic surges of the hormone far beyond what the human kidney would ever release at one time. And flooding the system like that triggers an entire cascade of off-target effects.
We are basically demanding that the body do something it isn't prepared for. And the drug acts on more than just the bone marrow, right?
Exactly that. The most immediate mechanical problem is blood viscosity. When you artificially cram that many red blood cells into the circulation, the blood literally thickens.
It's the difference between asking your heart to pump water through a network of tiny pipes versus asking it to pump molasses.
That's a perfect way to put it. The friction, the resistance, it puts immense strain on the cardiovascular system. Pumping that molasses damages the vascular walls over time.
Wow.
But the drug also directly activates platelets, making the blood much more prone to clotting.
Well, that's a bad combination.
It's terrible.
Yep.
So you have thicker blood, pumping harder, with sticky platelets ready to form a clot at the slightest provocation. And beyond the cardiovascular system, researchers eventually discovered that certain tumor cells actually possess EPO receptors.
Wait, really?
Yes. So in cancer patients, flooding the body with this powerful survival signal was inadvertently telling the tumors to thrive and proliferate.
That is horrifying. You are trying to cure the patient's fatigue, and you are accidentally feeding the tumor. Or turning their blood to sludge.
Yeah. And the medical field learned this reality through a series of landmark clinical trials that completely shattered that more is better illusion.
So walk us through those trials.
Well, the first major turning point was a 1998 study led by Dr. Anatol Bazarov, known as the normal hematocrit trial.
And just as a quick definition for you listening, hematocrit is simply the volume percentage of red blood cells in the blood. It's just another way of measuring what we've been calling the hemoglobin level.
Exactly. So Basarov's team took over 1,200 hemodialysis patients who already had underlying cardiac disease. They divided them into two groups. One group received standard ESA doses to keep them moderately anemic, which was the standard practice.
Okay.
The other group received massive doses of ESAs, specifically calibrated to push their blood to near normal levels.
That'll happen.
The trial had to be halted prematurely.
Wow. Stopped early.
Yeah, the data monitoring committee saw a terrifying trend. The group pushed to near normal hemoglobin wasn't experiencing a better quality of life. They were experiencing a significantly higher rate of deaths and non-fatal heart attacks.
Stopping a major trial early. I mean, that is a massive shock to the system. It completely contradicted the intuitive belief of thousands of doctors.
It was a deeply uncomfortable finding, and it honestly took time for the medical community to fully accept it. But the signal only grew stronger with subsequent research.
Like the Choir trial, right?
Yes. The Choir trial in 2006 looked at kidney disease patients who were not yet on dialysis and found the exact same correlation, targeting higher hemoglobin led to more cardiovascular events and deaths.
And then came the TREAT trial in 2009. This one focused on diabetic kidney disease patients using darbepoetin alpha, which is a longer-acting ESA.
Right.
And pushing their numbers to normal didn't just fail to improve their survival rates, it literally doubled their risk of suffering a stroke compared to the placebo group.
And at the same time, oncology trials focusing on breast cancer and head and neck cancer were returning grim results too. Hitting higher hemoglobin targets with ESAs in these populations actually correlated with worse tumor progression and shorter overall survival times.
So the accumulation of all this data forced a massive reckoning. The FDA finally intervened and completely reversed a decade of established medical practice.
They mandated a boxed warning, which is the strictest regulatory warning available.
Right, dictating that doctors must use the absolute lowest dose of ESAs possible and only to avoid a blood transfusion. The era of chasing a normal number was officially over.
And that regulatory shift fundamentally changes how we view the drug. It is an incredibly potent tool for artificially overdriving the body's oxygen-carrying capacity.
And once we acknowledge that power, it completely explains why this exact same biological mechanism was ruthlessly exploited outside the hospital.
Oh, absolutely.
We have to shift gears and look at how this drug completely reshaped professional sports. Because if you followed professional cycling or marathon running in the 90s and 2000s, this period is infamous as the EPO era.
Yeah, it was everywhere.
This is the specific chemical cheat code that allowed Lance Armstrong to dominate the Tour de France and eventually get stripped of his 7 titles.
But the context of sports doping often causes some confusion here. When people hear about an athlete doping, they default to thinking about anabolic steroids, right?
Right. They picture a drug designed to build massive muscle bulk or create this state of aggressive superhuman strength.
But ESAs do absolutely none of that. Injecting EPO will not help you lift a heavier weight or build a larger bicep.
No, endurance athletes weaponized it because it specifically elevates aerobic endurance, or VO2 max.
Exactly. When you're pedaling a bicycle up the French Alps, the limiting factor isn't muscle strength, it's the amount of oxygen your blood can deliver to those muscles before they suffocate and fatigue.
So by packing the blood with artificially high levels of red blood cells, a cyclist can sustain a grueling high wattage pace for an hour longer than the clean athlete next to them.
It was the perfect biological crime, honestly, because of a massive lag in testing technology.
Undetectable.
Virtually undetectable before a direct urine test for recombinant EPO was finally implemented around the year 2000. Because athletes were injecting a synthetic version of a hormone the human body already produces naturally, anti-doping agencies couldn't easily distinguish between the two.
But the side effects were still there. The athletes were pushing their hematocrit to dangerous, sludgy levels.
Oh yeah, sometimes even having to wake up in the middle of the night to exercise just to keep their thickened blood from clotting in their sleep.
That is insane. But, as terrifying as the sports doping era was, there was this entirely different, quieter controversy happening simultaneously within the United States healthcare system that is arguably much more troubling.
You're talking about the financial scandal.
I am.
Yeah, the financial scandal surrounding ESAs is a textbook case study in the dangers of perverse economic incentives in medicine. For years, the reimbursement structure for dialysis care in the U.S. operated on a fee-for-service model.
Right.
In practice, this meant that Medicare and private insurance reimbursed dialysis clinics separately for the ESA drugs they administered.
Meaning the clinics essentially purchased the drug at a discount, billed the government for the administration of the drug, and kept the spread as profit.
Yes, the drug itself became a massive profit center. Precisely that mechanic. The more ESAs a clinic pumped into its patients, the more revenue the clinic generated. Investigative reporting, highlighted by a major 2012 series by the Washington Post, exposed how this fee-for-service loophole incentivized aggressively high dosing.
Clinics were financially motivated to push patients' hemoglobin levels higher and higher, generating literally billions of dollars in drug revenue.
And the truly chilling part is the timeline here. This profit-driven push for massive dosing was happening concurrently with the BSARB, Choir, and TREAT trials.
So clinics were incentivized to administer the exact supraphysiologic doses that researchers were proving caused heart attacks, strokes, and tumor growth.
Yes. It eventually forced Medicare to radically change how they pay for dialysis, moving to a bundled payment system just to remove the financial incentive to overprescribe the drug.
It really highlights a critical vulnerability in healthcare. Whenever a payment structure directly rewards the volume of a drug administered, clinical decisions can be easily distorted, sometimes operating completely independent of the emerging scientific evidence.
It is a sobering reality check, for sure.
It is. Well, before we wrap up the biological story of ESAs, I want to run through a quick rapid-fire segment to clear up a few lingering misconceptions about this drug class, because the mechanics are still widely misunderstood by the public.
Okay, let's do it.
First and foremost, ESAs are not synthetic blood.
No. They contain 0 actual blood components. There are no red blood cells in the vial. The drug is purely a chemical messenger that instructs your own bone marrow to do the manufacturing.
Okay, second, they are completely useless in an acute emergency. If someone is bleeding out from a traumatic injury in the ER, administering an ESA will not save their life.
The biology simply doesn't move that fast. As we discussed, rescuing a precursor cell from apoptosis is only the first step. It takes 8 to 10 days for that single cell to mature,
Right.
meaning it takes anywhere from 2 to 6 weeks for the drug to meaningfully elevate a patient's hemoglobin levels. It is strictly a tool for chronic management, not acute blood loss.
Got it. And finally, the drug cannot perform magic in a biological vacuum. You cannot inject someone with an ESA and expect their anemia to resolve if they lack the basic raw materials required for construction.
The primary raw material for hemoglobin being iron. If a patient's iron stores are completely depleted, flooding the marrow with survival signals won't produce functional red blood cells. The body literally cannot build the protein.
So successfully using ESAs requires vigilant co-management with iron supplements.
Exactly, to ensure the marrow has the supplies it needs to actually respond to the drug.
Well, taking a step back to view the entire landscape, why does the story of ESAs matter to you? I mean, they represent a multi-billion-dollar specialty drug class that fundamentally transformed nephrology. Today, they're actively keeping hundreds of thousands of dialysis and cancer patients off of dangerous chronic blood transfusions.
But beyond their clinical utility, they serve as one of the most profound cautionary tales in modern evidence-based medicine.
They teach a hard lesson in clinical humility. The core premise was perfectly intuitive. If severe anemia causes suffering, then forcefully correcting that anemia back to a normal state must alleviate that suffering.
But rigorous scientific trials proved that perfectly intuitive assumption was dangerously wrong. We learned that while a biological mechanism can produce incredible benefit when replacing a deficiency, forcefully overdriving that same pathway can unleash devastating harm.
Which leads to a final thought I want you to mull over. We mentioned earlier that the kidney senses low oxygen using the HIF pathway, the hypoxia-inducible factor. Well, pharmacological science never stops evolving.
It does not.
There is a brand new class of drugs entering the market called HIF prolyl hydroxylase inhibitors, or HIF-PHIs.
And the delivery mechanism is entirely different. These are oral pills rather than synthetic injection.
Exactly. Instead of injecting laboratory-made EPO into the bloodstream, these pills work by inhibiting the specific enzymes that normally break down the HIF protein when oxygen is present.
So, by blocking those enzymes, the drug essentially tricks the kidney into believing the body is at a high altitude.
Right, artificially mimicking a low oxygen state. And this stimulates the body to ramp up its own endogenous EPO production naturally.
It is a brilliant pharmacological hack, but, it raises a massive lingering question that researchers are grappling with right now.
Yeah?
We know that injecting massive synthetic doses of EPO to hit normal targets caused cardiovascular damage and blood viscosity issues, while triggering the body's internal endogenous pathways managed to bypass those cardiovascular risks.
Or is human physiology about to teach us, yet again, that artificially overdriving our natural biological limits always demands a toll, regardless of how we flip the switch?
It's the million-dollar question.
It is definitely something to critically consider the next time a headline announces a flawless new medical miracle. And to think, this sprawling multi-billion-dollar saga, the lives saved from transfusions, the tragic clinical trials, the massive cycling scandals, all of it stems from a stubborn biochemist, a theoretical hormone, and 2,500 liters of 1970s urine.